Here we go. Okay, so we were talking about haplotypes, and these are unique sequences that normally exist in only one copy per person, unlike the vast majority of your genes, which exist in two copies per person because you've got two of each chromosome. You're diploid. Haplotypes are sequences that are found on mitochondria, which are inherited only maternally, and Y chromosomes, which are inherited only paternally. If you have a Y chromosome at all, not everyone does, you got it from your father's 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 ... father. And if you've got mitochondria, and everybody has those, you got them from your mother's 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 ... mother. The reason this matters is that you only have one haplotype per person, so you don't get genetic recombination. Hopefully you remember enough about mitosis from 1440 to remember that in meiosis, the chromosomes pair up, homologous chromosomes get together and then swap bits, right? Crossing over. That's genetic recombination. And it means that, for example, your own, say, chromosome 1, you might have got one of your copies of chromosome 1 from your mom, but when she made the egg cell that would become part of you, the chromosome 1 that she passed on was a cut and paste job of her parents' chromosomes number 1. And if you have kids, what you will pass on to them is a copy of chromosome number 1 that is a cut and paste job of your mom's chromosome 1 and your dad's chromosome 1. So every chromosome that you pass on is a cut and paste of your two copies. That doesn't happen with mitochondrial DNA, and it doesn't happen with Y chromosome DNA. The only thing that changes a haplotype is mutation. There is a fairly small but not zero probability that any given piece of DNA is going to mutate between one generation and the next. So your Y chromosome DNA, if you're a guy, is not necessarily an exact clone of your dad's. There may be some mutations on there that mean it's a little bit different from your dad's, and your dad's might be a little bit different from your granddad's, and his might be a little bit different from great granddad's, and so on. And the same thing is true of mitochondria as well. So that's the reason why we don't all have identical mitochondrial DNA and why all men don't have identical Y DNA. Those differences can be, for our purposes, we can make two types of differences, SNPs and STRs. An SNP is a single nucleotide polymorphism, and that's just a way of saying that at one position along the Y chromosome sequence, one person might have an A and another person might have a G. So at position 12,136 on my mitochondrial DNA, I might have a T, and Jenny might have a C, and Rachel might have a G, for all I know. They're just single nucleotide variants. They're also sometimes called UEPs, unique event polymorphisms. The other type of haplotype that we look into are single tandem repeats. And you might recall that there's areas of your genome where instead of having genes that code, you have stretches of DNA that are anywhere from 2 to 6 nucleotides repeated over and over. So there might be some point in my DNA where I've got ATGC, 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 ATGC repeated for a bit. Those are single tandem repeats. And at some position on my Y chromosome, I might have 7 repeats of ATGC, ATGC, and some other guy, Joe Biden, for all I know, might have 9 repeats, and Jason Momoa might have 13 repeats, and I don't know, my wife's uncle might have 6 repeats. Doesn't really matter. The point is that different people can have different numbers of single tandem repeats. These are important in forensics, by the way, because single tandem repeats are a major part of the CODIS database that the FBI collects on people. When you do DNA fingerprinting, you can match a DNA sample to a person based on the numbers of single tandem repeats at points all through the genome, not just the Y. This is a map of some single tandem repeats on the Y chromosome. They're just given code numbers, DYS 453, 446, 454, 459, yeah, they're just given numbers. The majority of these, by the way, don't actually do anything. There are some cases, like, for example, the gene for Huntington's disease, where too many STRs can actually cause defects in gene function. But for the ones that we're talking about here, they don't actually do anything. So they're not really subject to natural selection because they have no effect on survival. I mean, at DYS 447, I might have 12 repeats, and Jason Momoa might have 15, and this has nothing to do with anything. It has nothing to do with any of our physical or mental features. It's just there. I don't know why I keep going to Jason Momoa. It was the first name that popped into mind, but whatevs. Whatever. And the cool thing about this is that you can take, DNA samples from people, and you can measure the different SNPs and single nucleotide polymorphisms and short tandem repeats that they've got, and groups of them make well-defined haplotypes that have been given code numbers. So, for example, mitochondrial haplotypes are coded A through Z, and each one of these letters just represents a particular set of SNPs and STRs. And when you measure this in populations from around the world, we end up with something like this. You can see that the world looks very, very colorful. There's no absolute barriers, really. But there are some parts of the world where certain haplotypes are more common than other haplotypes. So, for example, in Europe, the most common mitochondrial haplotype is one called haplotype H. That's this light blue color that you can see makes up roughly half of the European population in Iceland, Britain, Spain, France, all of this area. In Southeast Asia, one of the more common ones is another set of haplotypes, another set of STRs and all that, that we call haplotype D. That's this dark blue. Some areas like, Central Asia here is just crazy diverse. Some areas are not very diverse. Haplotypes A, B, C, and D by far predominate in populations, native populations of North and South America. So we're all different, and you can use this to try to look for clues to genetic history. For example, Native Americans are said to have reached North America at least, the old answer was 13,000 years ago. They now think there may have been some that have been there for even longer. Um, there's some new evidence there that's really interesting. But the standard explanation is that they migrated across the Bering Strait, that they migrated from Asia across this way, possibly through land, possibly by boat. It doesn't really matter. From there to here. Okay, is that possible? Well, certainly. Do we know where they might have come from? Well, notice this purple right here. That's mitochondrial haplotype A. Any population in Asia that's got a high frequency of haplotype A is a plausible candidate for common ancestry with Native Americans. So where do we get haplotype A? HA right there, that's Han Chinese. So is that. Where else do we get that? KE right there. That's a group called the Ket people. Where else? We have some purple in here in Japan, and HT right there. That's Han Taiwanese. Okay. What else do we have? We have this bright red over here. That's haplogroup B. Well, all the ones that I've circled have haplogroup B. We have some more here. This is Chinese as well. We have some more red over here. That's Taiwan Aboriginals. I think I can't see right now whether we've got red right there. We've got some haplogroup. Haplogroup D is this bright blue, and we see some here, and we see some there. Haplogroup C is this kind of magenta, and we see it here, and we see a lot of it up here among the Yevenks and Yakuts. So we can start identifying plausible relatives of Native Americans from a time before they reached North America. There was an old theory going around called the Solutrean hypothesis, which stated that Native Americans actually crossed the Atlantic based on similarities in stone tools. But you'll notice that most of the haplotypes in mitochondrial DNA that are so common in North America aren't really that common over here. We have some of this purple haplogroup A, but we have haplogroup H over here in Western Europe, and we have virtually none in native North America. So we can rule out certain hypotheses for where the people came from. This is what it looks like with Y haplotypes. You can see that in Native American men, far and away the most common one is haplotype Q. That's this light purple. And there's several Asian populations that have high frequencies of haplogroup Q, including one over here, which I can't clearly see because there's a toolbar in my way, but there's a population called the Selkoops. It was right there. Dang it. Okay, it was right there. It is. There's the Selkoops. That has a high frequency of haplogroup Q. This is neat because the Selkoop population actually speaks a language that has some similarities with native North American languages, specifically Navajo and Apache. So not only languages, but genes just might be telling us a very similar story, that the ancestors of Native Americans, or at least Native American men, tended to come from this part of the world, from Northern Asia, Siberia. So that actually works quite interestingly. And we can do a little bit better than this. This was found about 10 years ago or so. Each one of those haplotypes that I mentioned can be subdivided into more specific varieties. So haplotype C on the Y chromosome, let's go back. Haplotype C is this kind of salmon colored thing. And you can see that there's salmon right there in some of these native populations have some of that salmon. That's Y haplotype C. And it's pretty common up here in Northern Asia, right? There's, a lot of it there. There's a lot of it there. There's some among the Han Chinese and so on. There's a subtype of haplotype C. It happens to be called C3 star. And it turned out, as people started looking more specifically at varieties of these haplogroups, haplogroup C3 star is shared by about 8% of all Asian men. You can see it's not particularly common in southern China or Laos and Cambodia and Thailand and Vietnam down here. But it's pretty common in northern China, in Mongolia. There are Mongol populations where 25% of all men have a C3 star Y chromosome haplotype. And it spreads all the way here to Central Asia, to Uzbekistan and Kazakhstan. And the Hazara are a people in northern Afghanistan, and they've all got this C3 star Y chromosome haplotype. Why does that matter? Remember that you get Y chromosomes only from your dad. So if two men have identical Y chromosomes, they must have the same father's father, right? They're either brothers or they share a father's father or a father's father's father or something like that, but they ultimately come from the same male common ancestor through a strictly male line of descent. What this means is the fact that some of these populations C3 star is so common means that everybody in these populations that has it must ultimately descend from the same great 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 grandfather. They must all share a patriline, a line of male ancestors that ends in a common ancestor. That made more sense in my head than it might be coming out of my mouth, but I hope you get the picture right here. So all of these people have the same great 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 grandpa, which raises the question of who great 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 grandpa actually was. And based on some estimates of how old this haplotype is, looking at mutation rates, he's been identified. The likeliest reason why 25% of the male population of parts of Mongolia have the same Y chromosome seems to be that they are all descended from a man named Genghis Khan. Genghis Khan was the creator of one of the largest empires in world history. He was the emperor of the Mongols. His armies of warriors on horseback conquered China, spread across Siberia, grabbed Central Asia, got all the way to Russia, were finally turned back at Egypt. But at one point, the Mongols ruled an awfully big chunk of the Asian continent, from the Middle East all the way to, they didn't quite make it to Japan. Incidentally, they didn't quite make it to Japan because they were about to invade on ships, and a typhoon, a hurricane-like storm, blew up and scattered all of their ships. And the Japanese considered that a miracle and called it the divine wind, or in Japanese, kamikaze. So that's where that word comes from. So how did Genghis Khan manage to spread his Y chromosome to so many descendants? Well, he is credited as saying the greatest joy for a man is to defeat his enemies, to drive them before him, to take from them all they possess, to see those they love in tears, to ride their horses, and to hold their wives and daughters in his arms. And unfortunately, I'm not talking about warm, comforting hugs here. Genghis Khan had 4 official wives. He had a very large number of concubines, kind of second-class wives. And there's no way to sugarcoat this. When the Mongols were on the march, they engaged in a hell of a lot of raping, like armies have throughout history. It's not a pleasant fact, and I apologize to bring that up, but it's something that happened. So Genghis had many wives, many concubines, and other opportunities for making descendants. He had 4 sons by his primary wife. This is them. And after Genghis died, okay, it says my internet connection is unstable, so I'm going to give it a minute. After Genghis Khan died, any Mongol that wanted to claim power had to prove descent from one of Genghis's 4 sons. That was called the Golden Lineage, Altan Urag. And I think the last male descendant of Genghis Khan, I think he died in 1920 or something like that. It wasn't that long ago. And if you were a man in the Altan Urag, you had lots of opportunities to get lots of wives and have lots of kids and leave lots of copies of your Y chromosome in future generations. And so we think by conquest and by polygamous marriage, Genghis Khan's Y chromosome is thought to have spread over an awfully big chunk of northern Asia. You can see that from, this part of Mongolia over here, not a very big region, he and his male descendants ended up ruling all the way to Russia over here, pushing into Georgia. And as I've mentioned, they finally got turned back in the Battle of Ain Yalut in what's now Egypt. They finally got stopped. And so we can get some insights, not always very pleasant or comfortable ones, but we can get some insights into events in human history by looking at haplotypes like this. All right, one moment. Hang on just for 1 second. I'm going to. Okay, there we go. Yeah, my wife was leaving the house, and we had to speak a few, sweet nothings and things like that that are really none of your business. Right. So that's the kind of thing you can do with haplotypes. Okay. Time check. Plenty of time. Plenty of time. With haplotypes, if you can sequence DNA, they're pretty straightforward because they exist in only 1 copy per person. It's trickier if you use most genes because with most genes, each individual has 2 copies. You have 2 copies of the vast majority of your genes, one from mom, one from dad. And just by looking at an organism, a person or a pea plant or what have you, you can't read the person's genotype because dominant alleles mask recessives. So this happens to be a purple pea flower, and it's purple. It's diploid. It's got 2 copies of each gene. Purple is dominant to white, but I can't tell if it's homozygous dominant or if it's heterozygous, not just by looking. Now, with incomplete dominance, of course, you can, as we did when I talked about these cats that were in the room, how you can tell by looking whether a cat has 0, 1, or 2 copies of the spotted allele, whether it's homozygous spotted, homozygous non-spotted, or heterozygous. So you can sometimes tell, but in many cases you can't. So here's a population of purple flowered peas. If every one of these flowers comes from a plant that is heterozygous, if every one of these flowers has the genotype big P, little p, then the frequency of the big P allele is 0.5. If every one of these plants comes from a homozygous dominant P plant, then the frequency of the purple allele is 1, because all plants would have the genotype big P, big P. Or it could be anywhere in between, depending on what portion of these flowers are homozygous dominant and what portion is heterozygous. We can't tell by looking. The frequency of the purple allele could be anywhere from 0.5 to 1. This is the last dead white guy I'm going to bring up for a while. Godfrey H. Hardy, mathematician at Cambridge University in Britain, did most of his work in extremely abstract fields, the kind of math that you really need to already have an undergraduate degree to really get into, a field called number theory, which I assume we teach it, but it ain't your father's algebra. Let me put it that way. He did most of his work there in this very rarefied, abstract sort of field. But a friend of his by the name of Reg Punnett, who he used to play cricket with, the 2 things that he loved were mathematics and cricket. Cricket is a British game that's kind of like baseball, but takes a lot longer, and has a flat bat instead of a round bat. Anyway, he used to play cricket with Reginald Punnett. This is the same guy who developed the Punnett square, by the way. He's that Punnett. And Reg happened to mention to Hardy that there was this problem of calculating allele frequencies. Now, a guy by the name of Udny Yule, one of the goofier names in the history of science, had claimed that Mendelian genetics was going to be a problem for evolution because dominant alleles would always increase in frequency in a population. Hardy sat down. The story is that he wrote a letter to the journal Nature. The story is that he wrote it over breakfast. The math is not remotely difficult. It's high school algebra. He probably, had the idea in about 2 minutes, and the rest of the time was spent jotting it down while waiting for his soft-boiled eggs. And he knocked off this letter in 1908 explaining what would happen. And it goes like this. Let's say we have a population with 2 alleles of one gene. We can expand it to what you get if there's more than 2 alleles of a gene, but let's not get ahead of ourselves. So we have 2 alleles of a gene, and we can call these big A and little a. And we can represent the frequencies of the alleles in the population as P and Q, lowercase p and lowercase q. Now, the frequencies we can represent as decimals between 0 and 1. If you want to turn them into percents, you can multiply by 100 and add a percent sign at the end. but we usually represent these as decimals. And the sum of P plus Q is always going to equal 1. If our population meets certain criteria, and I'll tell you what those are in a minute, then in that population, the genotype frequencies will be the homozygous big A. The frequency of that genotype will be the square of the frequency of the allele. It'll be P squared. The frequency of the other homozygote will be the square of that allele's frequency, Q squared. And the frequency of heterozygotes will be 2pq. You might have noticed that if P plus Q is equal to 1, then P squared plus 2pq plus Q squared is also equal to 1. And if that will be true if our population fits certain criteria. And if those criteria are met, the genotype frequencies and the allele frequencies will stay constant from one generation to the next. There won't be any change in allele frequencies or in the frequencies of genotypes. And this situation is called being in Hardy-Weinberg equilibrium, because a German guy named Wilhelm Weinberg came up with the same idea at about the same time, and two people, tend to share the credit. We usually call this Hardy-Weinberg equilibrium. If you are really patriotic, you might note that an American came up with the same idea pretty close to the same time, and sometimes he gets commemorated by people calling this Hardy-Weinberg-Castle equilibrium, but I'm just going to go with Hardy-Weinberg equilibrium. So when a population is in Hardy-Weinberg equilibrium, the following criteria hold true. The population has to be large. Technically, a population can't be in true Hardy-Weinberg equilibrium unless the population's infinite. And of course, we have no infinite populations. So any real world population will deviate a tiny amount, but it won't deviate significantly as long as it's a large population. And I'll talk later about just how large has to be. The second criterion is we can't have selection going on. Every individual in that population has to have equal probabilities of passing their genes on. No selection. No mutation must be going on. Now, in the real world, mutation always does happen, but in real world populations, the rate of mutations is so small that we can usually ignore it. The fourth criterion is that mating has to be random. And I should mention that when I say random mating, I'm not talking about, some frat party mixer gone horribly wrong or anything like that. I don't mean what back in the 1970s they used to call key parties. Look it up if you don't know. By random mating, what you mean is that individuals are not choosing each other based on genotype. What's the example I always think of? Okay, your eye color is determined by genes, so is your hair color. Okay, your hair color is genetically determined. Let's say we have a population of people that consists purely of observant church-going Baptists, and all of the men marry their high school sweethearts and settle down, and everybody is faithful, and nobody cheats except in country music songs on Kissin' 96. So I've just described Valonia, I think. You could still have random mating right there, and what it means is that the blondes and the brunettes have equal probabilities of mating. If the blondes are fighting off husbands with a stick, and the brunettes are all sitting at home and doing whatever you do if you can't get a date in Valonia, which makes me shudder, you would have non-random mating. If the brunettes are, you know, all have their pick of the suitors and the blondes are all staying home, you would have non-random mating. But if the odds of you getting to have kids have nothing to do with whether you're a blonde or a brunette, then you would have random mating, even though everybody is perfectly monogamous and faithful and all of that. So random mating doesn't really mean that you're picking your mates out of a hat. It just means that having a particular combination of genes gives you no advantage in finding a mate. There's no sexual selection. Inbreeding is another way that random mating is often violated. If you're mating with close relatives, or in some species, if you're self-fertilizing, that violates the random mating criteria as well. And then the final criterion is there's no migration in or out of the population. If a population ticks all those boxes, or at least comes very close, then you're not going to get change in allele frequencies over time. The allele frequencies are going to stay right where they are. The frequencies of the different genotypes are going to stay right where they are. Evolution's not going to happen. That's important because that gives you a null hypothesis. If you study a population and you find out that it's not in Hardy-Weinberg equilibrium, that's important because it means that at least one of those 5 criteria is being violated. Either the population's too small, or there is selection going on, or there is mutation, lots of it going on, or mating is not random, or there's migration in or out, or possibly some combination of those. So what you can do is use Hardy-Weinberg to make a prediction for a population and then compare it with what you observe in the real population itself. And if your prediction does not match up with the allele frequencies that you actually see, that means something interesting is happening. That means evolution is taking place, and now you have a reason to try to find out why it might be taking place. That may not be perfectly clear, but once I start working some actual problems, hopefully you'll see how this plays out. And I'll just mention that Hardy wrote an autobiography called A Mathematician's Apology, which was his attempt to convey to the general public what it's like to be a mathematician and think on an extremely high level of abstraction. And he was kind of proud on working on pure mathematics that didn't have anything to do with the real world. As he wrote, "I have never done anything useful. No discovery of mine has made or is likely to make the least difference to the amenity of the world." Notice the date on that. He's saying that right as World War II has kicked off, and right as Britain is starting to get bombed by the German Air Force, the Luftwaffe, and right as many of his friends are working for the war effort to develop radar, build better bombs, crack German codes, things like that. Hardy, I think, was kind of proud on working on math that you couldn't turn into a machine for killing people. Ironically, first of all, some of his discoveries about prime numbers have actually been put to use in internet security. You're able to send your credit card number to Amazon and have less than a 50-50 chance that it's going to get stolen by hackers in Russia. You're able to do that in part because of Hardy's work in number theory. But what's more, his best known achievement in biology is this very practical, very useful piece of elementary algebra that he probably didn't bother to even think about when he was writing his autobiography. And after our next break, I'm going to show you how it works. I'm going to move away from slides and I'm going to start working some of the practice problems that are up there right now on Blackboard. Uh, this might be this might be a good time for you to download these if you're watching this or following along. So I'm going to go ahead and stop recording now.